This video is for The Living Earth, Unit 3.5, Evidence for Evolution. Today's essential question is, how do molecular sequences provide evidence for evolution? Molecular sequencing, which is the sequencing of DNA or proteins and amino acids, can be used to analyze the molecules of life in groups of organisms. At the molecular nucleotide or amino acid level, organisms from bacteria to pigs have many similarities. All living things carry their genetic code in DNA or RNA. There are also the molecules that pass on traits to offspring in every living organisms. Many genes and proteins are also homologous across the living world. For example, one particular protein, cytochrome C, which functions in cellular respiration, is very similar in all living cells. In another example, bacteria and eukaryotes are very different kinds of organisms, yet both share the same genes that code for the ribosomal units used in protein synthesis, which suggests that bacteria and eukaryotes shared a common ancestor long ago. By comparing sequences in proteins or DNA of different organisms, scientists can determine how closely related they are. Generally speaking, the greater the number of differences, the greater the amount of time that has elapsed since they had a common ancestor. Molecular sequencing is just another piece of evidence that supports Darwin's original idea that with modification through natural selection, life evolved from a common ancestor. Molecular clocks. Molecular clocks are techniques that use genetic mutation rates to determine historical time when divergence of species occurs. There is some evidence that some evolutionary changes take place at the molecular level in a clock-like fashion. Over millions of years, random mutations build up in any given stretch of DNA at a fairly constant rate. This makes changes in DNA a powerful tool for analyzing and evaluating the evolutionary histories of related organisms, which enables scientists to calculate the dates when evolutionary lineages split. For example, imagine that a length of DNA found in two species differs by 2 bases, and we know that this entire length of DNA changes at a rate of about 1 base per 10 million years. That means that 2 DNA versions differ by 20 million years of evolution. Since each lineage experienced its own evolution, the 2 species must have descended from a common ancestor that lived at least 10 million years ago. When comparing different organisms, the more genes that are compared, the more accurate the dating.
Evolution results from changes in DNA. These changes are caused by random mutations, changes in the DNA sequence. Mutations are often detrimental, but some mutations are neutral. Neutral mutations neither benefit nor decrease an organism's ability to survive and reproduce. Neutral mutations are evolutionary changes and occur in a clock-like fashion. They act as a sort of molecular clock. Molecular clock is a technique used in evolutionary biology that estimates the age divergence of organisms from a common ancestor by looking at differences in their DNA. Scientists measure the differences between the DNA in certain genes. Often, mitochondrial DNA of organisms is used because its rate of mutation is well established. They use this molecular clock to determine when organisms diverged from a common ancestor. In this example, 2 different species had a common ancestor sometime in the past. To determine when they diverged from a common ancestor, scientists use a molecular clock based on a gene that they know changes by 2 bases every 25 million years. They observe that the species differ in the DNA of this molecular clock gene by 4 bases. If the species differ by 4 bases, they must have diverged from a common ancestor around 50 million years ago. It's difficult to calibrate the molecular clock against actual time because the rate of mutation depends on factors such as generation time and population size. The calibration leads to some uncertainty in dates of divergence. However, using multiple genes along with fossil data can help narrow down the dates. Scientists have identified genes that have more constant rates of change than others. These make the best molecular clocks. Biologists have used the molecular clock technique to determine the date of the divergence of humans and chimpanzees. The data show that the last common ancestor of chimps and humans lived around 5 to 7 million years ago. This ancestor evolved into various hominid species, one of which is an ancestor of modern man. In 2008, researchers found DNA in Neanderthal fossils from this cave in Croatia. The scientists compared the mitochondrial DNA of humans and Neanderthals to estimate divergence of our modern lineages from these ancestors. Molecular clocks are an imprecise but useful tool. The technique has been used to investigate important issues, such as the origin of modern humans.
At this point, you should be able to generate a full and appropriate response to today's essential question. How do molecular sequences provide evidence for evolution?